Good morning, students. Today we will study the next part of human health and diseases. So what are the topics involved? Let me see here. So we'll study the types of acquired immunity and structure and types of antibodies. So as you have studied that there are two types of immunity, innate immunity and acquired immunity in the previous lecture. So today we will see the types of acquired immunity. So two types of acquired immunity, cell mediated immunity and humoral immunity. I explained their definitions in the previous lecture. Now today we will study in detail these immunities and their mechanism. Then the structure of the antibodies, they are known as the immunoglobulin chemically, and their types. So we'll see here that types of acquired immunity. So there are two types of acquired immunity. The first immunity, which is known as the cellular immunity or cell mediated immunity, and the second type of immunity, that is the humoral immunity, or we call it as the antibody mediated immunity. So this is cell mediated immunity or cellular immunity or cellular response, you can say, and humoral immunity or antibody mediated immunity. So we'll see this cellular immune response or cellular immunity. What happens here? So here the types of cells which are involved in this cellular response or cellular immunity, they are the T cells. So response of T cells to the antigen we will study here. T cells respond to antigen by producing a clone of T cells, means the many copies of the T cells are formed. Each T cell recognizes a specific antigen. Therefore, the body contains separate T cells for every antigen that it encounters. So when a specific antigen is encountered in the tissue, these T lymphocytes, they are programmed to recognize the particular antigen, return to the specific T lymphocyte domains of the lymphoid tissue, where they transform into the lymphoblast. So these T cells, they are programmed specifically to recognize the particular type of antigen, and they are being activated, transformed into the lymphoblast, and lymphoblasts then divide mitotically and produce the activated T lymphocytes. These activated T lymphocytes, they migrate into the circulation, enter into the circulation, migrate to the site of antigen, and then they show their activity. So major the types of activated T lymphocytes are there. These are the cytotoxic T cells, that is CTL, TC, that is cytotoxic T cells are there. These helper T cells are there, and the third cells which are known as the suppressor cells or the memory cells also. So let me understand their function. This cytotoxic T cells, these cells cause direct destruction of antigen containing cells by release of lysosomal protein. So these cytotoxic cells, they release the that substance that is lysosomal proteins. So they directly destruct the antigen containing cells, and cytotoxic destruction is particularly important in dealing with the viruses which are otherwise relatively inaccessible within the host cell. So they kill the infected cells by releasing these lysosomal proteins. Then we will see the helper cells, helper T cells. These cells, they play rather an indirect role in stimulating the B cells, B lymphocytes. Indeed, helper T cells are essential in initiating most immunological responses, exerting their influence by a variety of highly specific regulatory factors. So that first function we have studied here of the T cells, that they stimulate the production of antibody by the B cells. So they help the B cells in order to produce the antibodies. Then helper T cells, they also produce a variety of known specific substances. Collectively they are known as the lymphokines. These lymphokines, they enhance the phagocytic effectiveness of local and blood-borne macrophages. So macrophages which are present in the blood and the area, local area, their phagocytosis increases under the influence of this substances that is known as the lymphokines secreted by the T helper cells. So helper T cells are specifically destroyed by the virus responsible for AIDS, resulting in the loss of immunity. So that you have studied about the AIDS, where immunity of the person decreases. Why? Because these helper T cells are destroyed by the HIV, that is a virus of AIDS. So person becomes weaker, means its immunity becomes weaker. Now the next is the suppressor T cells. Some of the cells, small proportion of the activated T lymphocytes, it remain in lymphoid tissue where it acts as a memory cells. And memory cells means they are keeping the memory of that antigen on which they attack. So these cells are capable of mounting a more effective secondary immune response on subsequent exposure to the particular antigen. It means this some of the activated cells, they change into which activated this T lymphocytes. So some of the T lymphocytes, they remain in lymphoid tissue and change into the memory cells. So these memory cells, they are capable to produce more effective secondary immune response when the second time exposure of that particular antigen takes place in the body. So that's why they are memory cells. Now next we will see here the this humoral response or humoral immune response or humoral immunity. Now this immunity is there, the response of B cells to the antigens. And B lymphocytes are involved in humoral immune response. Immunocompetent B lymphocytes, each programmed to recognize one particular antigen only, and they are released in the general circulation, from which they feed the lymphoid tissue, mainly lymph nodes and spleen. And in contrast to T lymphocytes, B lymphocytes do not continuously recirculate through the body, but rather they make contact with the antigen and processed by the macrophages. So they attack on antigen, they are being in contact with antigen directly, and they are taken up and processed by the macrophages. And once they are being activated, these B lymphocytes, they transform into the प्लाज्मा ब्लास्ट, और ये प्लाज्मा ब्लास्ट डिवाइड होकर के ये फॉर्म करती हैं एंटीबॉडी प्रोड्यूसिंग प्लाज्मा सेल्स. सो देयर इज ट्रांसफॉर्मेशन ऑफ दिस that B cells into the plasma cells. So first B lymphocytes transform into the that plasma blast, and plasma blast, they transform into the plasma cells which produces the antibody. Understood here? Now these plasma cells, it produces antibody, and these antibody now attack or bind themselves to the particular antigen, and these antibodies bind to here or more antigens simultaneously and make them immovable. So here antigen cannot move. Such immovable antigens are then phagocytosed, means by phagocytosis they are killed by the macrophages. So understood children here? So macrophages are also here means working along with the B lymphocytes. As I explained you here already, that immunocompetent B lymphocytes programmed to recognize particular antigen, and further they release the antibodies, and these antibodies, they further attach with the antigen, bind with the antigen, and this immovable antigen is phagocytosed by macrophages. So macrophages, they kill this antigen by phagocytosis, means by engulfing them. Macrophages, these are the monocytes. Now antibodies can also act as opsonins also. Opsonin means they can coat the antigen. so that antigen becomes much tastier to the macrophages, and this process is known as the opsonization. So this is the humoral response. So in this way we that see that acquired immunity, how does means this acquired immunity work in our body? There are two ways: cellular immunity and the humoral immunity. So understood? In cellular immunity no antibody is produced, but in humoral immunity antibodies are produced by the lymphocytes. Cellular immunity or cellular response, it is caused by the T lymphocytes, and humoral immunity, it is caused by the B lymphocytes. Here one more important function of that T helper cells or T4 cells also, they are known as this T4 cells. They actually help this B cells. So B cells can bind the antigen directly, but they require the help of this helper T cells for its proper activity. So in this manner they help the B cells. T cells, they cannot bind directly to the antigen, but B cells, they can bind to the antigen directly, and this T cells, T helper cells, actually they help the B cells in their proper activity. So understood, children? In this manner this immunity takes place in our body, means this is the way that immune system works. Now next we are going to study the antibodies. What are antibodies? Antibodies are actually glycoproteins. They are termed as immunoglobulins, IgS, which are produced in response to an immune reaction and specifically bind to antigens responsible for initiating the reaction. So antibodies, they are the chemical molecule glycoproteins, and they are termed as immunoglobulins. The short form is IgS, which are produced due to the response of our immune reaction, means antigen enters into our body, and due to that reaction that antibody is produced. So in response to an immune reaction they are produced, and these antibodies specifically bind to the antigen responsible for initiating the reaction. So various types of antibodies are there: IgG, IgE, IgD, IgM and IgA. So let me study here the structure of antibody. So here this diagram you learn well. Diagram is most important in this question, that is structure of antibody. So all antibodies, they contain two identical light chains and the heavy chains. So this is light chain and this is the heavy chain. So two light chains are there and two heavy chains are there. So all antibodies consist of two identical light chains and two identical heavy chains. So these are heavy chains and these are the light chains. Now these chains are held together by a tetramer by disulfide bond and form a Y-shaped structure. So this disulfide bond is holding the chains together. Now each chain is divided into two regions. These regions are the constant region and the variable region. You can see here the constant domain and variable domain. So you can call it as constant region and the variable region. Now where is this constant region and variable region? Let me study here. So constant region of light chain, as I said you, this is light chain. Constant region of light chain, which is known as the Cl. You can see here, it is written here, Cl. Constant region of the light chain is known as Cl, and it is towards carboxyl terminus. You can see here the carboxyl group is present. So constant region of light chain, which is called as Cl, it is towards the carboxyl terminus. And variable region, you can see here Vl. Variable region is of light chain is called as Vl, and it is towards amino terminus. You can see here the amino group is there. So it is towards the amino terminus. Now let me see the constant region of heavy chain. So this is heavy chain. So constant region of heavy chain. These two are the heavy chain, these two are the light chain. So constant region of the heavy chain is CH. You just see here it is written here CH. So constant region of the heavy chain CH, it is towards the carboxyl terminus. And variable region of the heavy chain, you just see a VH. Here also you see the VH. So constant, this variable region of the heavy chain, here you see the VH, it is towards the amino terminus. So understood, children, here that the two regions, constant and variable regions of light chain and heavy chain. Now the portion of the antibody that binds the antigen is the variable region. So here you see the variable region is actually the place where the antigen binds. So here you can see antigen binding site. So portion of the antibody that binds with the antigen is the variable region, that is amino terminus. At this place antigen binds. And when an antibody molecule is treated with the enzyme papain, the antibody is cleaved into two fragments. So by enzyme reaction, the antibody cleaved means it breaks down into two fragments. What are the two fragments? FAB. This FAB is the antigen binding fragment, and FC, this is the crystallizable fragment. So AB, that is the antigen binding, and C means crystallizable. So FAB, that is antigen binding fragment, and FC, that is the crystallizable fragment. Now light chains are further of two types. These light chains are of two types. They are kappa and lambda. A given antibody can have either 2 kappa or 2 lambda, but never a mixture of both. So this is the structure of antibody. You learn it through the diagram. Now let me see the types of antibodies. So there are these 5 types of antibodies. These type, the type of that heavy chain, it determines the type of antibodies. And on this basis, there are 5 types of antibodies. These are IgG, IgM, IgA, IgD and IgE. So that different types of heavy chains are there. On that basis, this different type of antibodies. So let me see further their functions. IgA. So what is the IgA? This immunoglobulin A, what is its function? Found in mucus, saliva, tears and breast milk, and it protects against the pathogen. So its main function is to protect the body against the pathogens. Then IgD. This IgD, it is the part of B cell receptors, and it activates basophils and the mast cells. So present on lymphocyte surface as the receptor and activates the B cell. IgE is protecting against the parasitic worms and responsible for allergic reaction. So they are mediator in the allergic reactions. Then IgG. IgG, they are secreted by plasma cells in the blood and able to cross the placenta into the fetus. And one more is there, IgM. That IgM, it activates the B cells. So all these are the functions of the different type of antibodies and present in the different that secretions. Now type of antibodies in saliva, serum and colostrum. This is given in your syllabus, so you just pay attention over here that this different type of antibodies present in the different secretions in our from our body. So let me see here that what type of antibodies are present in saliva. So in saliva, this is IgA. So each immunoglobulin is specific in function, and about 70% of the normal human serum is IgG. So serum contains all these 4 types of antibodies in them except this IgD, and this IgG is 70% in the normal human serum. It is most common type of antibody. This IgG, it passes from mother to fetus through the placenta and provide immunity to the newborn. While here further this IgG is the workhouse of the immune system. Then the 7% of the human serum is IgM. Each IgM molecule is composed of 5 monomeric units, and it activates the complement systems. IgA, now let me see IgA. IgA accounts only about 2% of the total serum, and it is found in relatively low levels in the blood, but it is the main immunoglobulin to be found in the body secretions like tears, then saliva, nasal dropping, colostrum, and the milk. So this IgA is found in all these secretions. The presence of IgA in colostrum and milk appears to be appears to be an important way to provide in the infant with immunity to disease. So that colostrum, that is the that first milk after the birth of the baby secreted, this breast milk contains this IgA antibodies in it, and it provides immunity to the infant. And only 1% of the serum, it is the that IgD, very less is there, and little is known about this serum, but it seems that it regulates the synthesis of other immunoglobulins. And Ig is also there, that Ig, that is 0.02% of the total immunoglobulin present in the serum, that is IgE, and when combined with the antigen, it shows the allergic reactions. So these are the different types of antibodies in the saliva, serum, and colostrum, and their functions. Now, two more terms you have in your syllabus, and their more detail you can see from the book, because about these terms you have studied in 10th class already in detail. Immunization, that is vaccination. What is immunization or vaccination? It is a way to trigger your immune system and prevent serious life-threatening diseases. So, triggering, waking up your immune system and prevent the serious life-threatening diseases. What is vaccination? It is the administration of antigenic material, means administration of vaccine to produce immunity to a disease, and vaccines can prevent the effects of infection by the pathogens. And the different types of vaccines are there. So there is a list of vaccine in your book. You can study these vaccines from your book also, that DTP, hepatitis B, oral polio vaccine, BCG for tuberculosis,